So we started this lecture last week, yeah? We got a little bit through it. Do you remember that? We talked about the first and second law of thermodynamics. Yeah? We talked about energy interconversions, yeah? What kind of energy we have here? Kinetic energy, yeah? We put a dam there, what happens to the water? It stops moving. Does the energy go away? No, the energy is still there. Yeah, the first law of thermodynamics tells us energy is never created or destroyed, but it can be interconverted. And it's been converted from kinetic energy to what? Potential energy. And then it flows through the turbine, and the turbine spins around and generates what? Electrical energy. And then that electrical energy generates light. Yeah? The second law, these interconversions are never 100% efficient. Do you guys remember discussing that? We pointed out that when you touch the light, especially if it's an incandescent light, what do you feel? When you put your hand on a light bulb, it feels hot, right? You can burn yourself with an incandescent bulb. You don't burn yourself with a compact fluorescent, but it does feel warm. And why? Well, because some of the energy that we were trying to convert from electrical to light didn't get converted the way we wanted it. And instead of getting converted from electrical to light, it got converted from electrical to heat. Heat energy is the most common form of lost energy. Does that make sense? Right? The energy doesn't get destroyed, but it's doing something besides what you wanted. We wanted to illuminate the room. We didn't want to heat the room with the light bulb. Does that make sense? Right? So every time there's an interconversion, there's a loss of efficiency. Some of the energy doesn't get fully translated in the direction we intend. Right? So water behind a dam, potential energy, yeah? Okay. Most chemical reactions solely involve the exchange of covalent bonding partners. We talked about this, yeah? We talked about exergonic and endergonic reactions, yeah? And we talked about how, which of these reactions is endergonic? The left or the right? Endergonic. The one on the right, yeah? That requires energy. This one is giving off energy, yeah? The explosion is the energy given off from the chemical reaction of hydrogen and oxygen. So going from the left to the right, energy is given off. From the right to the left, energy is required. We need a battery or some kind of electrical power to make all these bubbles form. And what's in the bubbles? Oxygen is in some of the bubbles. What's in the other bubbles? Hydrogen, yeah? Hydrogen and oxygen are both formed from the electrolysis of water, yeah? Okay. So, which molecule pairs have more energy in their chemical bonds? We talk about this, yeah? What's the answer? A, of course, right? Gasoline has more energy than carbon dioxide, right? Oxygen, when used to burn the gasoline, allows the release of a lot of energy, and that energy is supposed to be translated into kinetic energy of a moving car, yeah? Does that make sense? Is there energy lost? Yeah, most of the energy is lost to heating the engine. We heat up the engine so much that we actually have a radiator, right, that sends cool fluid into the engine to cool it off, yeah? So it doesn't overheat, right? Right? So the majority, 75% of the energy in the gasoline is wasted, yeah? Kind of a shame, but people have certainly been trying for a long time to make car engines more efficient, and this is how far they've gotten so far, right? What about human cells? How efficient are they? Where are the high energy molecules, on the left or the right? The high energy molecules are on the left. Sugar has calories, yeah? Does carbon dioxide have calories? No calories here, yeah? Water has no calories. That's why the bubbles in your Diet Coke don't provide any calories, right? There's a big advertisement on the front of that soda pop saying no calories, but there is carbon dioxide in those bubbles, yeah? You agree? Yeah? So, no calories here, calories here. And the calories go into the cell, and what does the cell use them for? Yeah, to making energy, right? What are these little sparky things supposed to be? These are ATPs, yeah? ATP, and again, look how well written that is. Again, I'm sorry it doesn't, things don't work so good, but anyway, how efficient is a cell? Not much more efficient than a car, yeah? About the same. Most of the energy in the sugar that we eat is wasted as heat, yeah? So just as engineers are trying to make more efficient automobiles, evolution has certainly been trying, certainly has a big evolutionary incentive to be more efficient with the food we eat, and yet this is as good as we've gotten so far. Okay, so according to collision theory, exergonic reactions happen on their own, but not without sufficient energy. We talked about this, yeah? You guys remember that? Right? So, does this graph show an exergonic or endergonic reaction? Understanding this graph is something that I will be asking you to do, yeah? So make sure that you understand graphs that are like this. It's an exergonic reaction. That's correct. Is that Whitney? How did Whitney know? Somebody besides Whitney explain how Whitney got the right answer that, that this graph shows an exergonic and not an endergonic reaction. Anytime you're ever looking at a graph, you want to look at how the axes are labeled, yeah? How is the y-axis labeled? It says energy, yeah. So E is for energy, yeah? How much energy do the reactants have? That much, yeah? That much. That much. Yeah? How much energy do the products have? Whatever it is, it's less, yeah? It's only that much, right? And the difference, there we go. The difference here, that represents the energy lost in the reaction, yeah? If this is hydrogen and oxygen, and this is water, this represents the explosion of the Hindenburg. Does that make sense? You guys get that? Right? That's the energy given off. What does this represent here? The EA. That's the energy added, the energy of activation. Yeah? EA, and I'll write this here because this board works a lot better than that gizmo. The energy EA equals energy of activation. In other words, what? The energy to get the reaction going, right? In this case, it was, in the case of the Hindenburg, I guess, it was the grinding of the metal tower on the metal frame of the Hindenburg to create the spark that caused the explosion, right? In the case of the Bunsen burner, right, it's the spark that we use to get that reaction going, yeah? Does that make sense? Right? It's the energy you invest in order to get this payoff, yeah? Right? This is the energy invested. This is the net energy return. This part, the difference between the investment and the return, is the profit, yeah? Does that make sense? In a money term. And I'm wearing a tie, so I know all about these things. Sorry. Anyway, so in order to have molecules react, they need to collide. They need to crash into each other, and they need to collide with sufficient force. The spark provides the accelerator, the accelerating energy to make them crash together with enough energy to get the reaction going. Okay, so ATP is the energy currency of all life. We've talked about this, yeah? So the chemical reaction, where do we get our energy? From food, right? I didn't have a bowl of ATP, a bowl of Cheerios, yeah? So the energy, but Cheerios have calories in them, yeah? And the calories in Cheerios come from the chemical bonds in the Cheerios, yeah? In the same way that the chemical bonds in the gasoline are what give the gasoline energy, right? It's the same atoms on the left and the right. The only difference here is the covalent bonds on the left and the covalent bonds on the right holding these molecules together, yeah? And so the energy has to be in the chemical bonds, yeah? Does that make sense? Right? So the energy in the chemical bonds of the Cheerios is energy that my body is going to use to do all the blabbering on that I do all day long, yeah? Right? And so that energy, when my mouth flaps away, the muscles in my lips and my tongue and so on that are flapping away are not having little explosions of Cheerios. They're having little explosions of ATP. Does that make sense? And I think I've said this a few times, yeah? So the food we eat goes into making ATP, and then the ATP is what makes our muscles move, what makes our brains work, et cetera, et cetera. Is that good? You guys good with that? All right. So how does this work? Well, it works from things called redox reactions, and we're going to skip redox reactions, okay? Skipping redox. Maybe I'll come back to it. All right? So as long as I don't talk about it, it's pretty complicated. Students have trouble with it, and the learning to time payoff is, you know, I think if you don't fully learn about redox, you won't be better off. It's better to know it than to not know it, but it might be better to know other things down the line. Does that make sense? You guys with me here? So it won't be on exams unless I switch course and say, oh, we've got a few minutes. Let me just explain this. Yeah? Is that clear? At least for now, it ain't on there. No redox. Yeah? And when it comes time for the exam, we always go through the study guide, and I scratch off things you don't need to know, right? That's what we did last time. That's what we'll do this time. And so you guys will know, buddy, we're not doing redox. You skipped it. Yeah? Is that clear? And this is being recorded. Yeah? Okay. All right. So the presence or absence of appropriate enzymes determines the metabolic character of microbes. We've talked about this before, yeah? Well, if that's the case, how do I end up breaking down the milk and the yogurt and the kiwi fruit and the what else was on that bowl, and the Cheerios themselves, and the rice checks and the bananas and the strawberries? It really was quite a. You should have been there. Man, that was the best bowl of cereal I've made all morning. Wait, it was the only bowl of cereal I ate all morning. Anyway, so in order to digest, can I digest all that stuff? Not all of it, right? In there was milk. And as I often expound upon, I don't digest lactose in milk very well, yeah? Because my body doesn't make lactase enzymes anymore. Likewise, there's fiber, right? And fiber is made out of cellulose, and I can't digest that, yeah? But all the other stuff, I can absorb, and I can convert to calories, to energy, to ATP, through chemical reactions involving enzymes. Does that make sense? So one of the most important chemical reactions is this one, respiration. Right? And all I did you guys copy this down before? You probably have it somewhere, but copying it down ain't going to hurt. Yeah? So, lipids are inspired for energy, but what we're going to look at is the respiration of this stuff here. And what is this? Glucose, carbohydrate. Yeah? You guys with me here? Okay. Questions? Guys all right? Okie doke. So, microbes like this can be classified according to how they obtain energy with which to make ATP. And so, this is how we do it, yeah? We do aerobic respiration. There it is again, right? So if you didn't copy this down, you could just write aerobic respiration over this, yeah? Does that make sense? This chemical reaction is aerobic respiration. This is the primary means by which humans gain ATP and the reason we breathe, yeah? Without oxygen is because when you don't have oxygen, you can't do this chemical reaction. And if you can't do this chemical reaction, you can't get ATP. And if you can't get ATP, you'll die because your cells will die. Yeah? Does that make sense? We breathe oxygen because our cells need oxygen. Yeah? And they need oxygen to do this to get ATP to stay alive. Guys good? Questions? Okay. So, but this picture, and have I showed you this freaky picture before? Well, what disease does this guy have? This is a soldier in one of the Napoleonic Wars, and he's dying of a disease. See all his muscles tying up? What disease? Every muscle in his body is tense, yeah? Look at his toes. They're all curled up. His fists, his jaw is clenched. His jaw is clenched. There's a little hint there. It's tetanus. Nicely done, Beverly. Yeah? We've talked about tetanus before, yeah? Lockjaw, yeah? This guy is dying of it. Well, what causes tetanus? Clostridium tetani, right? Clostridium tetani is an obligate anaerobe. In other words, a bacterium that will die in the presence of oxygen. You with me? This guy will die in the presence of oxygen, yeah? So, do you think he's doing the same chemical reaction that we're doing? The way we get ATP, right? The way we get the energy essential to life. All life uses ATP, yeah? All life has to have energy, yeah? And all life uses ATP as its energy currency. But not all life does this, yeah? How could an obligate anaerobe like Clostridium tetani do a chemical reaction that requires oxygen gas, right? If the oxygen gas is going to kill them, they can't do this in order to get their ATP. You agree? Well, they have to be getting their ATP somewhere. How are they doing it? Well, the lecture that we've got right now is going to expound upon how microbes that use oxygen get their ATP and how microbes that don't use oxygen get their ATP. Is that clear? All right, and I'll try to go through it as quickly as possible and trim down all the stuff you need to know and all the stuff you don't need to know. But here it is. Glucose and oxygen form carbon dioxide and water, but the primary reason this is important is because it's an exergonic reaction that yields energy that is used to make ATP, and much of the energy is lost as heat. Yeah? Guys good? All right? So, aerobic respiration is the most basic means for cells to acquire energy. Are there calories in protein? Yes. Are there calories in fats? Yes. So calories and fats can also be burned for energy. Yeah? Why are we focusing on carbohydrates? I guess it's mostly traditional. Yeah? Does that make sense? And it's and it's fundamental, yeah? It's common. Humans do it. Most microbes do it. So that's where we're starting with from the base of this reaction. Is that clear? But don't forget, right? Lipids have calories too. Amino acids have calories too. And if we want to burn those, we can burn those alternatively to sugar. Is that clear? All right. So this is the same reaction as throwing wood on a fire, right? Wood is made out of cellulose. Cellulose is a carbohydrate. And it burns very well, doesn't it? Yeah? Okay. But we control the reaction, right? We don't have our sugar that we eat doesn't catch fire. We break it down and dismantle it piece by piece. And we're going to skip this question because it's about redox. Okay. The powerhouse of the cell, we can answer this one, as we learned about chapter 4. Powerhouse of the cell. Come on, don't let me down. You guys learned this in high school, right? I just reminded you. What is it? Mitochondrion. Good, good. Okay. So, and why is that? It's because the mitochondrion provides most of the cell's ATP. Yeah? Okay. So, here is aerobic respiration. The reaction that we saw is a very, it looks simple enough here, and it is pretty darn simple here. The way living things do it is quite complicated. There are 3 major steps: glycolysis, citric acid cycle, electron transport. There's an intermediate step that sort of counts, but let's look at this. If you remember this, this is the primary stuff you need to know. Glycolysis. Glucose is split. 2 pyruvates are formed. You get a little ATP, 2 ATP per glucose that you break in half. Yeah? Citric acid cycle. I keep thinking I fixed this. Pyruvates are broken into CO2. Redox molecules are NAD+ are charged up. So we're not going to talk too much about redox molecules. A little ATP is gained. 2 ATP per glucose in the citric acid cycle. And then electron transport chain, lots of ATP is made by ATP synthase. What is in ATP synthase? It has the suffix ASE, so it must be an enzyme, yeah? ATP synthase, what kind of job does ATP synthase do? Trick question. Synth, what does it mean, synth? Synthesize, yeah? So ATP synthase synthesizes ATP, makes ATP. It's a money printing machine, yeah? Is that clear? All right. And it makes a lot of ATP per glucose, yeah? Okay, so again, if you know this, you know a good 50 to 60% of what I'm expecting you to know, yeah? The other thing I'm going to stress is the mechanism by which electron transport works. Is that clear? Everything else is just details. All right, you guys with me? All right, so here they are. Glycolysis, right? So glycolysis, can we break down that word? Glyco means glucose or sugar or something like that, yeah? Lysis, what does that mean? To break, yeah? So breaking sugar, taking sugar and breaking it in half, yeah? So that's step 1, a little ATP. Step 2, citric acid cycle, a little ATP. Step 3, electron transport and ATP synthase, lots of ATP. Okay? Got it? Questions? Okay. So glycolysis. So here's glycolysis. Here's what's going on here. What's this molecule here? Glucose. This is not the way we usually draw glucose. Usually when we draw glucose, we see a little hexagon, yeah? Isn't that the way we usually draw glucose? It's drawn sort of bean counting wise. What are these 6 little balls? They're carbon atoms. They're the 6 carbon atoms in glucose, yeah? Here they are, C6, yeah? What are the lines connecting the carbon atoms? Those are the covalent bonds holding the molecule together. Is that satisfactory? So what's happening? We're breaking the molecule in half, and here, this covalent bond is being broken, yeah? The one right in the middle, yeah? That covalent bond, what is a covalent bond? Do you guys remember what covalent bonds are? When electrons are shared, yeah? So it's a shared pair of electrons. Excellent, Kimberly. Yeah? So this thing called NAD+ is actually doing a redox reaction. You don't need to worry about that. Picking up these 2 electrons. These 2 electrons, this is an oversimplification, but you could think of them as these 2 electrons here. Yeah? Does that make sense? And it's picking up these 2 electrons, and it's going to take them, guess where? There's 3 steps, right? Glycolysis, citric acid cycle, and electron transport chain. So these things are going to be taken to the electron transport chain. Okay? Is that clear? So a lot of what we're doing when we do aerobic respiration is stripping down glucose for. Is that clear? Guys good? Questions? Okay. This simple graph, or it's not that simple, yeah, but as not that simple as this picture is, it's actually way more crazy complicated, yeah? 10 steps, right? First, glucose turns into glucose 6-phosphate, and then to fructose 6-phosphate, and then to fructose 1,6-bisphosphate, and then half of it turns into dihydroxyacetone phosphate, yada, 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 yada. There's 10 steps in this process, yeah? Why do I show you these steps? You certainly do not need to know them, yeah? The reason I show you is just because what makes each of these steps happen? An enzyme makes every step happen. Each step is catalyzed by an enzyme. So glycolysis, this process, actually involves an enzyme pathway, right? In other words, 10 guys on an assembly line, yeah? Or I guess this is really a disassembly line. We're breaking the molecule in half, yeah? It's like this is a big molecular chop shop. Do you guys know what a chop shop is? Raise your hand if you do know what a chop shop is. Raise your hand if you're like, what? Why do you use these analogies when nobody knows what they mean? So a chop shop is where your Honda Civic goes to die, yeah? The people who steal your Honda Civic, you'll never see it again. Why? Because they dismantle the whole thing. They take every part apart, and then they sell all the parts one by one. Does that make sense? Likewise, glucose is going to get stripped down piece by piece, and all the pieces are going to be sold for energy. Does that make sense? Okay, so hopefully that analogy is useful for people who know what a chop shop is. And hopefully you never learn any more about chop shops. Yes? Pyruvate. Yeah, so pyruvate, that's a good question. Basically, what you need to know about what pyruvate is, it is the 3 carbon, it's half of a glucose. It's pyruvate, you could call it pyruvic acid, but basically it's what's left over. It's a little more oxidized. It's a 3 carbon molecule that used to be half of a glucose molecule. All right, and here's what happens to pyruvate. Pyruvate, here are the 3 carbons. Everybody look up at the 3 carbons of pyruvate, held together with covalent bonds. And again, in covalent bonds, we have electrons, and those electrons have energy, yeah? So we break this one off. The covalent bonds, the electrons, some of them are picked up to form NAD+ into NADH. And this thing, again, this is the same NADH we saw before. NAD+'s whole job is to pick up electrons and take them to the electron transport chain. Guys got that? Right? So here, it's picking up 2 electrons, and it's been reduced, right? The charge has gone down from NAD+ to NADH. That means its charge has gone down by 1, from +1 to 0. And the electrons did that, yeah? Okay. Okay, so the Krebs cycle. Questions? All right, here we go. So the Krebs cycle is basically where the rest of the covalent bonds in the pyruvates that we got from glycolysis get stripped down also, and the electrons, 3 NAD pluses, turn into NADHs, 1 FAD, whatever the heck that is, gets turned to FADH2, and again, these molecules here are stripping down electrons, and those electrons, you can think of it as that they were holding the carbons together. Now that the carbons are no longer held together, they float away as little carbon dioxide gases. Okay? Is that convincing to you? Yeah. Yeah, so what do we call this thing? This says citric acid cycle equals Krebs cycle. That's another name for it. Equals TCA cycle. Equals tricarboxylic acid cycle. Right? So the textbook, I think, uses, I can't remember, I think it uses Krebs cycle. Yeah? And that's why in my big letters here, Krebs cycle, in the figures I ripped from other textbooks, they use other terms, the citric acid cycle. But it's the same thing. Yeah? These are all synonyms. Yeah, Whitney. What do you think? Every time we breathe in, what happens? We take in oxygen, yeah? Every time we exhale, you guys learned this in physio class, right? Everybody learned this in physio class, yeah? You breathe in, oxygen goes in. You breathe out, carbon dioxide is released, yeah? Where is that carbon dioxide coming from? Your body doesn't make it by magic. It's making it by the same process, right? We take in oxygen and we release carbon dioxide when we do respiration in black, yeah? Guys with me there? Questions? All right. All right, next step, electron transport. Questions? You guys tired? Oh, that's good. It gets harder. So this step is the hardest one. It's also the one that I think is the most interesting. It's also the one that I want you to know the most about. So grit your teeth right now. Right? Electron transport, in which the electron transport chain generates a proton gradient. Do you know what a proton gradient is? Do you know what a gradient is? You know what protons are. What kind of charge do protons have? They have a positive charge, yeah? What is a gradient? A gradient is a disparity, a difference. It is, you ever driving and you're going down a hill and there's a picture of a truck on a yellow sign and it says 6% grade. Raise your hand if you've seen a sign like that, right? That means you're going down a steep hill, yeah? So a gradient is, in that case, the grade or the gradient there is the difference between the high hill and the low valley. Does that make sense? The proton gradient means that you've got a lot of protons in one place and not so many in another place. And as it turns out, there's a cell membrane or there's a membrane in between. Yeah? Does that make sense? And we'll see that very shortly. But you should know the term gradient. Yeah? It is, yeah. So this is a concentration gradient of protons. High concentration of protons on one side, low concentration of protons on the other. Yeah? Right? So what about the charge on electrons? Positive? No, negative. Yeah, good. Right? So electrons are negatively charged, protons are positively charged. The electrons in the electron transport chain create a proton gradient. Okay? Guys good? And then ATP synthase, what does it do? Makes ATP. It makes tons of it, and we'll see how that's done. Okay? All right. So here is the energy graph of the electron transport chain. Here is NADH and FADH2. Do you remember these molecules? We just saw them, right? What did they do earlier on in glycolysis and in the Krebs cycle? They pick up electrons, yeah? And what are they doing? This arrow shows that they're handing electrons to these purple things, yeah? These purple things are proteins, yeah? And there's energy in the electrons, and this is the energy graph. These guys, the electron transport chain, is transporting the electrons from this guy to 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 this thing down here, which is oxygen. Yeah? You guys with me here? You guys got that? So the electrons go to oxygen, and then oxygen, when it gets electrons, turns into water. Right? It gets what's called reduced into water. All right, so electrons flow in, and then you might think of these as little water wheels, yeah? And the water wheels spin around and generate energy, yeah? Is that good? All right, so let's look at more detail about how that works, right? Here are the guys that we just saw, still drawn in purple, the proteins in the electron transport chain. Where are they? It looks like they're membrane proteins, yeah? They're sitting in a membrane, and we're familiar with transmembrane proteins, yeah? Well, these ones, their job is to grab electrons, yeah? So, imagine I'm a protein in the electron transport chain, yeah? And these represent electrons, yeah? What happens to me when I pick up these two electrons? I become negatively charged, yeah? And if that happens, what would a proton think if it saw me walking down the street? It would be attracted to me, yeah? Because I have a negative charge, yeah? Does that make sense? Guys with me? Yeah? So I could lure that proton toward me, and then as soon as it came up to me, I could grab it and I could throw it on the other side of the membrane I was sitting on. Does that make sense? Once I did that, I could hand the electrons off to another protein in the electron transport chain. And as soon as that, you didn't even ask to be part of this. You're like, I was just sitting here minding my own business. Sorry about that. So, Jessica, do you mind picking up the electrons? Thank you. So, Jessica is another protein in the electron transport chain. I have just handed her the electrons. What happens to her charge? It becomes negative. And what do protons think of her? Attracted to her, chemically attracted to her, yeah? Right? She can grab one of those protons and put it on the other side of the membrane, yeah? Off to another member of the electron transport chain. Thank you. Say it again. How many proteins? Well, 1, 2, 3, 4. It sort of depends on how you count them, right? Because these proteins are big and complicated. It's a few, yeah? It goes, the best answer is that the electrons go all the way through. And they go to the very end, and at the very end, right, when almost all the energy, so now Jenny is protein 3 in the electron transport chain, yeah? And protein 3 becomes negatively charged, and then Jenny can grab a proton and put it on the other side of the membrane. Does that make sense? Right? And then once she does, she passes off the electrons to the next member, and then the next member is protein 4, and and protein 4 will attract a proton. Protein 4 will grab that proton, dump it on the other side of the membrane, and then will, instead of passing those electrons off to another member of the chain, we'll just pass them to oxygen, yeah? And then oxygen will take them, and that will create a vacancy for more electrons to be added to the chain. Does that make sense? But each member of the chain that holds electrons grabs protons and puts them from one side of the membrane to the other, yeah? Protons are positively charged, right? So there's a lot of, I draw H plus here, and I draw H plus because of the H is for hydrogen, and the nucleus of a hydrogen atom is just a single proton, yeah? Does that make sense? So that's why H plus means a proton. Guys with me here? Right? So we get a high concentration of protons on one side, a low concentration of protons on the other. Right? Well, what is this used for? This is used for, this is effectively stored potential energy. Do you see the relationship between this membrane and the dam we saw earlier in the lecture? The dam holds back water. What is this phospholipid bilayer holding back? Protons. Good, good. Everyone see that? So, what does this thing look like then? We can see protons flowing back through it. We can see a little arrow going around in circles, and we can see ATP being generated. What is this thingy right here? This is a protein. This is a special, very special protein. This is ATP synthase. So let's watch a little video about ATP synthase. I think it's pretty healthy and helpful. No, keep recording, and we can watch this. The internets are kind of slow as usual. Always got to wait. Yeah, I guess I was recording because I thought that I would be talking about chapter 6. And because my Sunday class was ahead of this class, when I would record my Sunday class and then put it on the internets so that you guys could listen, yeah? Correct? The problem is that on Sunday, yesterday, this microphone ran out of batteries. So now, if I'm going to record chapter 6, it's up to you guys because you guys you're not in the lead, but you're in second place in terms of material covered. ATP, synthase. Here we go, first hit. All right, so this is pretty good. Everybody watch. We'll turn up the volume.
ATP synthase is a molecular machine. ATP synthase is a molecular machine that
Fiddlesticks. No, the last one. There we go. Thank you.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized.
So you see what's going on here? Right? So this thing is smashing ATPs, yeah? ADP, yeah? ADP is coming in with a phosphate. ADP plus phosphate is being smashed into ATP. What makes that happen? This thing, yeah? What gives this thing the energy to assemble the ADP and the phosphate? The flow of the protons, yeah? What does this thing remind you of? It works like a, what did they say? It's a molecular machine that works like a turbine, yeah? A turbine is the thing that spins. It's like the water wheel, yeah? A 20th century water wheel, yeah? How long have we been making things like Hoover Dam with a hydroelectric generator at the bottom of a really big dam? Maybe Hoover Dam was what, 1935, 1936, yeah? So this is how long has this ATP synthase been around? This thing is older than eukaryotes, right? The reason that eukaryotes swallowed up mitochondria for the endosymbiotic theory, right? You guys remember the endosymbiotic theory? We talked about that. The reason is because the little bacteria that got swallowed up had this thing, right? And the eukaryote wanted it, right? Why did it want it? Because it made its energy output go up by 10 times, yeah? The total amount of ATP in a glucose, for every glucose, we can get about 36 to 38 ATP. And of that, 32 to 34 is from the electron transport chain and ATP synthase. Yeah? So again, without this, how much ATP can we get? Hardly any, yeah? Don't you think it's remarkable that this is a molecule, yeah? This thing evolved, and it evolved billions of years ago, and yet it was only in the 20th century that people got the ability to make devices at the macro scale that work just like it, yeah? This thing is working just like a hydroelectric generator in a hydroelectric dam, yeah? Isn't it? I think that's pretty cool, personally. Right? So, this is like the generator. Where's the water? Where's the dam? What's the dam? The phospholipid bilayer. It's not a cell membrane. It's the inner membrane of a mitochondrion, or it could be the cell membrane of a bacterium, yeah? That has ATP synthase, yeah? And where's the water? Instead of water, what do we have? We have protons, yeah? Flowing through, right? And they flow through, and it's the power of the flowing protons makes this thing spin around, and as it spins around, every rotation, 3 ATPs get made, yeah? Pretty remarkable, yeah? One other question. Can you see a similarity between the way ATP synthase works and some other cellular machinery we've talked about in class that prokaryotes have? Anything spinning around in a membrane like this that you can recall? That thing, yes, that is correct. It's that thing. And that thing is the part, it's in the cell membrane, right? It's the flagellum, yeah? Right? That spins around, and as it spins around, the bacterium gets to move, yeah? If you look at the DNA, right, this is a protein. The blueprint to make a protein is in the DNA. If you look at the DNA sequence of ATP synthase, and you look at the DNA sequence of the flagellar motor, right? Very similar DNA sequences. common evolutionary origin between them. Yeah? That's a great question. Let's look at that and get that answered. Did you guys hear the question? So, who is doing aerobic respiration? We do it, yeah? Humans do it, right? And humans are eukaryotes, and eukaryotes have mitochondria, yeah? So, this could be, what do we see here? What are these purple things here? What is that? These are proteins in this membrane. This is the electron transport chain, yeah? What's this blue guy here? This is ATP synthase that's spinning around to make ATP. You guys see that? Guys with me here? You see the relationship between this picture and the video we just saw and the previous pictures in the previous slides in the lecture, yeah? All right. So, where is this? Well, it looks like it's in the inner membrane of the mitochondrion, yeah? This membrane here is the inner membrane of a mitochondrion. Does that make sense? Guys with me? Right? So, are mitochondria the only things that have ATP synthase? No, plenty of bacteria got them, right? Bacteria had them before eukaryotes ever showed up, yeah? So, if we're talking about a bacterium, well, a bacterium has an inner cell membrane, and that's where the ATP synthase and the electron transport chain sit in many bacteria. Does that make sense? Bacteria that can use oxygen, aerobic bacteria, or at least facultative bacteria. Guys got that? Okay. Questions? Okay. Moving on. So, in aerobic respiration, oxygen is the final electron resting place in the chain, right? Here's the electrons. They get handed, right? This one shows in a little better detail, yeah? This is from a more basic textbook, but it's still showing pretty good detail. NADH gives the electrons to the electron transport chain. They go through, and then they come out to oxygen, and then oxygen gets reduced, yeah? Does that make sense? So oxygen pulls the electrons through the chain, so to speak. All right. So ATP bookkeeping. How many ATP for glycolysis? 2. How many ATP for citric acid cycle? 2. How many ATP for electron transport chain? Tons. 30 odd, yeah? Is that clear? So, again, the vast majority of ATP comes from the electron transport chain, right? So, again, where do these things happen? This table answers in more detail Whitney's question. In a eukaryotic cell, glycolysis happens in the cytoplasm. The Krebs cycle happens in the mitochondrial matrix, and the electron transport chain is in the mitochondrial inner membrane. In a prokaryotic cell, glycolysis happens in the cytoplasm. Krebs cycle also happens in the cytoplasm because there are no mitochondria in a prokaryotic cell, yeah? And the electron transport chain happens in the cell membrane because there's no mitochondrial inner membrane, yeah? What if it's a gram-negative? You think it's the outer or inner membrane of the gram-negative cell? Probably the inner, yeah? Does that make sense? Guys with me here? Okay, phew. This is pretty hard, yeah? During cellular respiration, NADH does what? Here's a hard question too, yeah? People saying D, is that correct? Good on you if that's the answer you gave. D delivers its load to the electron transport chain. So, is reduced to form NAD+? That answer is wrong because, well, we didn't really talk about redox, yeah? Is biochemically converted into ATP? No, it helps to make ATP, but it doesn't transform itself into ATP. Does that make sense? Our food is the energy to make ATP, but it doesn't become ATP. Does that make sense? Okay. All right. Good. So, does that mean that if we run out of oxygen, that we can't do anything? Yeah? Human cells can do glycolysis faster than human lungs can take in oxygen, yeah? So here's Ronald McDonald. He's doing the bench press, yeah? So do you guys know what the bench press is? It's kind of like doing push-ups, only fancier, yeah, and more macho, I guess, or something. I don't know. But what happens when you do push-ups? How do your arms feel? Sore, yeah? So sore is kind of a special kind of pain, right? It's not the same, it's not the same as all the other kind of pain you get, right? Sore means you feel like a burning sensation, yeah? Have you ever heard somebody on the exercise bike say, "Come on, man, feel the burn. Feel it burn." Is that what your spin instructors tell you to do? Feel the burn? Raise your hand if somebody's told you in your life to feel the burn before. Right? So, what is the burn? Lactic acid. Your overworked muscles. What do you think? How many people have heard of lactic acid? Lactic acid buildup in your muscles, yada, yada, yada. Right? Feel the burn. Right? So, when people are exercise junkies, they know about this stuff, yeah? When you are pedaling a bicycle up a hill, your muscles in your legs burn, yeah? Does that make sense? And that's because in your muscles, lactic acid is accumulating, yeah? What about these guys? Right? Are these guys, again, these guys, right? Clostridium tetani, the thing that causes tetanus, is not doing aerobic respiration. Furthermore, humans can max out their ability to do aerobic respiration. Yeah? So, Leb, you have a question? So what about anabolic steroids? Yeah? Is that the question? So anabolic steroids are basically what they do. Here's a cell, right? An anabolic cell is a cholesterol derivative, yeah? Are molecules that look the most like cholesterol, and cholesterol sits in your cell membranes. As it turns out, this is a chemical. Really, what steroids do is they don't improve your ability to process lactic acid or do aerobic respiration or make mitochondria. What they do is they go into your cells and they go into the nucleus of the cell and they turn on genes that make your body do stuff. Yeah? So they turn on genes for actin and myosin in your muscles, for hair growth in your face, for making more certain brain chemicals in your brain. Yeah? And that's how you get a roid rage in your brain. That's how you get extra facial hair, and that's how you get bigger muscles. Does that make sense? But basically, what steroids are doing is affecting the genetic expression of your cells, not affecting the physiology of the cell in the short term. Does that make sense? All right. So here's a guy running Boston or something, yeah? And he's conked out because the lactic acid in his muscles has caused his muscles to fail, yeah? What's going on here? Here we have microbes, yeah? In barrels, along with some grape juice or some barley malt or something, yeah? What kind of microbes are in there? Yeast, yeah? And is there oxygen here? They're certainly eating food, but there's no oxygen in these barrels, yeah? So what do? Do they make lactic acid? They actually don't make lactic acid. I don't buy a bottle of wine because I want to get lactic acid. Let's just put it that way. What's in here? Alcohol, right? So, for those microbes and for those living things that can't do aerobic respiration, right? Either because they're maxed out in their ability to do aerobic respiration or because they just don't because they're obligate anaerobes, they have to do something else, an alternative to aerobic respiration. Again, respiration is common, widespread. We do it, but it's not universal. Not every living thing on Earth does it. Yeah, Juan Keys. Yeah, so some anaerobic, yeah, so I'm getting to that, yeah? So if I don't answer that in the next 5 minutes, you let me know, okay? All right, so here are some options. One is anaerobic respiration. Some microbes can use an electron transport chain, but instead of giving the electrons to oxygen at the end, you understand what I mean by giving electrons to oxygen at the end, yeah? Instead of doing that, they give the electrons to something else, something besides oxygen, like nitrate or sulfate, yeah? And so that is one thing. Humans actually can use their electron transport chains for oxygen and nothing else. So if there's no oxygen around, we can't do anaerobic respiration. What we do is fermentation. And for fermentation, the only source of ATP is glycolysis. How much ATP do you get from the process of glycolysis? 2. So we're going, when we are building lactic acid, we are going from 38 ATP for every glucose down to 2 ATP for every glucose. Is that clear? Is that, you guys understand? All right. So, and we are doing lactic acid fermentation, yeah? So fermentation is any spoilage of food, any process that makes alcoholic beverages. We define fermentation as is glycolysis, no oxygen, no Krebs cycle, no electron transport chain, just glycolysis. Right? So here is respiration. Glycolysis, Krebs cycle, electron transport chain, yeah? And ATP generated all along the line. Fermentation, glycolysis only, and then some way to basically dump the electrons. NADH needs to dump the electrons on the electron transport chain. If there's no electron transport chain, it'll just dump them somewhere else. And one place it can dump them is alcohol and make booze. This is alcohol fermentation, right? Here is glycolysis, here to here, and then here is where fermentation happens, and the electrons are basically dumped back onto the molecule. Carbon dioxide is where we get the bubbles in our champagne and so on. We don't make alcohol, we make lactic acid, yeah? Questions about this? I'm sort of ripping through some things. Here is something that I want to describe, right? The reason I'm talking about this is because fermentation is a diagnostic tool, right? So there are many end products of fermentation produced by many kinds of microbes. Yeast, when they do fermentation, they make alcohol, and we can get our beer and wine and booze, yeah? But other bacteria, if we're eating yogurt, right? Lactobacillus is in yogurt. How does yogurt taste if you have plain yogurt? It's kind of sour, yeah? So they're making lactic acid, yeah? The tartness of yogurt is from the fermentation of lactose sugar into lactic acid. So these guys, yeast and yogurt microbes, are doing the same thing. They're doing fermentation, but they're not making the same waste product. The waste product of yeast is alcohol and booze. The yeast product of yogurt microbes is lactic acid that makes your yogurt tart, yeah? There's lots of other fermentation products, and I'm out of time, but just give me 1 more minute, okay, please? All right, so many end products of fermentation, including lactic acid. How about you ever ferment kimchi, sauerkraut, delicious funky cheeses. Raise your hand if you like to eat stinky cheese. One person. Two people, because I like to eat stinky cheese, yeah? Right? So all of these different microbes can make all kinds of crazy flavors in fermented food. And that's because fermentation is very diverse, yeah? And again, the important thing is that fermentation allows us to do fermentation tests, yeah? These tests show different chemical reactions based on different ways in which microbes ferment various carbohydrates. Is that clear? You guys got that? Any questions? All right, that's enough to go. Wait, one keys. All right, what's your question one more time? Alcohol? To lactic acid? Do all anaerobic bacteria produce lactic acid? Answer, definitely not. Some anaerobic bacteria make lactic acid. Others make propionic acid. Others make butyric acid. This smells horrible. This is Clostridium. Clostridium tetani is one Clostridium. Clostridium perfringens causes gangrene. And if you've ever smelled gangrene, right, the smell of rotting flesh, right, it smells horrible because of some of the compounds that Clostridia produce, yeah? E. coli produce alcohol, lactic acid, succinic acid, acetylene, formic acid, hydrogen gas. All of these things are very, very different, yeah? So to answer your question, do all of them make lactic acid? Definitely not. There's alcohol and a bunch of other stuff too. Does that make sense? Got it? And if you have more questions, ask them. All right, Chapter 5, Part 1 is going to be on that quiz, yeah? See you guys Wednesday. Hasta, you know, Wednesday. Miércoles, yeah. Gracias. All right. And stop.